Good afternoon. I think this is one of the most talked about studies at this year's WCLC, along with the Tropion 2. And this is a molecule which is in development in China by Akiso Biopharma, which has now been acquired by an American company. And when you see the presentation on the 8th of September, their stocks were lying at about 43 Hong Kong dollars. And after the presentation, it shot up to about 90 Hong Kong dollars within 2 days. And currently, it's doing somewhere about $63 for a share price. No, I didn't buy the stock. It's not yet listed on the NSE. So this is an interesting molecule. AK112 is a bispecific molecule which targets the PD-L1 as well as the VEGF, and they looked at going head on with King Keytruda, because we know that in October 2016 Pembro did get the approval in first line metastatic NSCLC, both as monotherapy and in combination with chemotherapy. So the bispecific antibody against the PD-1 and the VEGF, this had shown promising clinical efficacy, and this was in their first phase 2 study with the AK112-202 study, and the HARMONi study, which was also known as the AK112-303, was a phase 3 study to compare the efficacy of Ivo with Pembro as first line therapy in patients with PD-L1 positive advanced NSCLC. The study design looked at enrolling about 388 patients and 264 PFS events, providing a 90% power to detect a hazard ratio of 0.67. And the interim analysis for the PFS was planned when 70% independent radiological review committee assessed PFS events occurred. So this is the study, stage 3B, stage 4 advanced NSCLC, patients who have not received any prior systemic therapy, no EGFR or ALK mutation-driven tumors, patients were selected. Patients were having good performance status of 0 or 1, and the mandatory enrollment was at least have more than 1% PD-L1 positivity. They had further stratified the population into based on stage 3B, C or 4, and histology-wise, whether squamous as well as non-squamous, because the phase 2 study did show that squamous population benefited with this drug much more as compared to non-squamous. The PD-L1 stratification as per TPS was more than equal to 50% versus those between 1 to 49, 398 patients. 1:1 randomization, which is a good thing to see now. There are very few trials which are doing this. Many are 2:1, which can skew the statistical analysis at times. Ivo in a standard dose of 20 milligram per kg every 3 weekly, 198 patients were randomized to this versus Pembro single agent 200 milligrams 3 weekly, about 200 patients. The primary endpoint being PFS, the secondary endpoint being OFS, and the PFS assessed by investigators. And both the treatments were continued until no clinical benefit or unacceptable toxicity or up to 2 years. The baseline characteristics between both these groups are well matched. When we see stage 3B, stage 3C, they were about only 8% patients. So you see here that predominantly 92% patients are stage 4, about 45% patients are squamous carcinomas, and PD-L1 more than or equal to 50% forms about 42% of patients. About 12 to 14% patients had liver metastasis at baseline, and about 16 to 19% patients had brain metastasis at the baseline. And when we look at the performance status, we clearly see here that probably there is a representative population of what we see in clinical practice, with almost 85-87% being PS1. In terms of age, 65 and above were about 50-55%. Now, this was the primary endpoint based on the independent radiology review committee, and the Ivo did score better over Pembro with an 11.1 median PFS versus 5.8, and a stratified hazard ratio of 0.51. Initially, they had started out with 0.67 in the clinical when they did the initial statistical analysis. So this turned out to be a statistical significant improvement, and there was a difference of about 5.3 months between both these study groups. When we look at the subgroup analysis, Of course, when we go by the stage first, the stage 3B/C form only about 10% of the entire population in each arm. So that's the reason possibly that it is crossing the midline. In stage 4, we clearly see a hazard ratio of 0.49 on the forest plot. Squamous versus non-squamous, no real difference. In terms of the TPS positivity, more than 50%, a hazard ratio of 0.48, 1 to 49% hazard ratio of 0.4. In terms of smoking status, also not much of a difference between both these groups. Age-wise also, whether patients are younger than 65 or they are more than 65 years of age, we see a hazard ratio is almost the same. Now, when we look at the subgroup analysis based on PD-L1 low and PD-L1 TPS more than 50%, these are the survival curves. And of course, based on the histology, the squamous as well as non-squamous, both are showing that there is a benefit of using this bispecific monoclonal antibody, which is targeting PD-1 as well as VEGF. In terms of the secondary endpoints, the overall response rate, disease control rate, and duration of response, 50% ORR versus 38, 89.9% disease control rate versus 70%, and median duration of response was not reached, and both ORR and disease control rate grossly higher than what single agent Pembro does. So safety summary was also discussed about these group of patients, and whenever we are using VEGF directed therapy in squamous carcinomas, we are all worried about the bleeding with our past experiences with bevacizumab in these group of patients. However, when we see in the squamous subgroup, more than equal to grade 3 toxicities seen in about 22%. Serious treatment-related adverse events in about 18%, which is the same in both these arms, and treatment discontinuation in 2 and 3% respectively. When we look at the most common treatment-related adverse event having incidence more than 10%, we are looking at proteinuria, liver enzyme derangement, bilirubin elevation, hypertension, which are common toxicities related to VEGF inhibition and also related to immunotherapies. The immune-related adverse events, when we are looking at more than grade 3 events, 7 and 8% in both these groups. Serious immune-related adverse events, surprisingly lower with the IVO group in 5% and higher with the Pembro. So I don't know what is the specific reason why this is happening, but possibly it may have to do with the selective binding of the PD-1 as well as the PD-L1. So when we look at specifically the VEGF related adverse events, proteinuria has been seen in about 31% of patients, hypertension in 15%, more than grade 3 in 5%, hemorrhage in about 14%, and these, as per the authors, the hemorrhage grade 3 which happened in 2 patients, they had non-squamous histology and was not reported in the squamous carcinoma patients. In terms of the quality of life C30 EORTC scale, there has been almost comparable time to deterioration of global health status in patients who have received both these therapies. So in conclusion, there was an improvement in the investigator radiological review assessed PFS in patients with advanced NSCLC and PD-L1, which was more than 1%. With the IVO as compared to the Pembro, 11.4 versus 5.8 months. The benefit was consistent across whether it was squamous or non-squamous and whether the TPS was more than 50% or between 1 to 49. There was a higher rate of overall response as well as disease control with the IVO. Overall survival data will mature over time, and the event-driven overall survival analysis will be reported. My take on this is that we are comparing a dual combination versus single drug, and we've seen the data. I know it's not a direct comparison, but you have the data of ABCP versus BCP, where addition of immunotherapy to the BEV and chemo combination has added another 5 months of PFS in that trial. So possibly, I do not know whether it is related to combining VEGF activity as well as PD-L1 and looking at 2 drug versus 1 drug here rather than just looking at single agent Pembro. So possibly the drug is good. We are looking at a good drug at our hands, and hopefully once we have the overall survival data, this drug does have potential to change practice. There was also some criticism about using this drug only in Chinese patients, and there are more trials going on across the Caucasians and in the US also now that this has been acquired by an American company. So they are looking at trials in different population subsets too. So hopefully we should have more answers in another few years. Thank you.
Thank you, Dr. Adar. Two comments, and I will request if you can have some opinion on that. One is that for PD-L1 1 to 49%, Pembro alone is usually not the standard of care. That is discussed as a drawback of this study. Do you agree that this is drawback or this is acceptable? What do you think?
So when you look at the phase 2 data, initially the Pembro had come in with all PD-L1 positivity more than 1%. So you do have activity of Pembro also, but when you look at Pembro chemo, it works better. So I would have been happy looking at Pembro and combination for that 1 to 49 subgroup, because that is the current standard of care. But obviously, the way the clinical trials are designed, we all know that they are done in a particular way with a particular intention in mind. However, what is heartening to see is even in that more than 50% group, this drug seems to be better.
So another point is that in squamous, we barely use bevacizumab because of the obvious reason of hematemesis hemorrhage. Any take on that? Because if you had used Pembro plus bevacizumab in the other arm, what would have been the results? Any take, because this is answer cannot be given.
This answer can't be, but as we have data from the Atezo-Bev, there are also we have seen that using it in non-squamous EGFR mutation-driven tumors, ABCP has not really increased the risk of hemorrhage in that subgroup of patients when you are using it on progression. So maybe there is a trial which needs to be designed in that way. If we have access to both the drugs, I think we should be the ones to look at that as a standalone phase 2 data. Maybe we can start as a collaborative group and collect our data to see where we are.
Thank you.